Welcome to the Discovering the Structure of DNA slideshow. In this slideshow, you will learn about the structure of DNA and you will learn about the discovery of DNA. What is DNA? DNA is also known as deoxyribonucleic acid. The deoxyribo portion of the word represents the sugar that is found in DNA. DNA holds all of the information in a cell. In a eukaryotic cell, it's going to be located in the cell's nucleus. In a prokaryotic cell, the DNA will be located in the nucleoid region. DNA also has a huge job. DNA's job is to tell a cell what proteins to make. We call that coding for proteins. So DNA codes for proteins that are essential to life. DNA is a nucleic acid. The monomer of a nucleic acid is a nucleotide. A nucleotide has three parts. It has a phosphate group. It has a sugar. In this case, the sugar is deoxyribose. It also has a nitrogenous or nitrogen base. If you look at the picture to the right, you will see how all three of these components are connected together. DNA has four nitrogenous or nitrogen bases. The first is thymine, which is abbreviated with the letter T. There's also adenine, which is abbreviated with the letter A. There's cytosine, which is abbreviated with the letter C, and guanine, which is abbreviated with the letter G. We like to call DNA a ladder. If you think of a ladder, it has sides and then it has rungs, which are the parts in the middle that you step on. The sides of the ladder in DNA are made of sugars and phosphates, and they alternate: a sugar, a phosphate, a sugar, a phosphate. Nitrogen bases, or my adenine, thymine, cytosine, and guanine, are going to pair together and make up the rungs, or the middle part of the ladder. This next bullet has a typo in it. The atoms within the two sides of the ladder are held together by strong covalent bonds. They're really hard to break. In the middle, the rungs are held together by weak hydrogen bonds between the nitrogen bases. DNA was discovered as part of a collaboration by several different scientists. The scientists that we're going to cover are Erwin Chargaff, Rosalind Franklin, James Watson, and Francis Crick. Rosalind Franklin is one of my favorite historical scientific figures. She worked with X-ray photography to help to try and find the structure of DNA. So she wanted to know what DNA actually looked like. She took a famous picture called Photo 51. I'm going to show you that in a moment, which revealed DNA structure. And this was in 1952. Rosalind Franklin died of cancer in 1958. We tend to think that that's due to her prolonged exposure to X-rays without any personal protective equipment. So it's actually pretty tragic that she died, and I'm going to tell you why in just a moment. So here's her famous Photo 51. Doesn't look like much to me, but she was so brilliant that she used this to defend and deduce the structure of DNA. Okay, so Watson and Crick are also pretty important historical figures when it comes to DNA. I'm sure you've heard of them before because they are actually credited with finding the structure of DNA in 1953. Only that's not really what happened. So James Watson got a peek at Rosalind Franklin's Photo 51, and he and Francis Crick used that photo with other evidence to help them describe the structure of DNA. What they said is that DNA is a double helix, and the strands are held together by weak hydrogen bonds formed between the bases. Unfortunately, this paper was published, and Rosalind Franklin didn't get any credit because she was already deceased at that point. It's pretty sad, and we're going to talk more about that in class. So make sure you get your notes complete for this. Remember that that is an assignment, and I will see you guys all tomorrow.